Hello and welcome back to Survey of Biology, and we're on Unit 10, Section 2, and we're going to continue our discussion of Mendelian genetics. And today we're going to explore kind of the differences between genotype and phenotype. So what we see on the surface of an individual is what we call their phenotype, and what their genes actually are, what's in their DNA, is called their genotype. And so what we're going to find out is the genotype, the chromosomes, the DNA that they have, are what codes for the proteins that will make up their phenotype, so their physical expression. Okay, so phenotype versus genotype. So an organism's phenotype is its observable characteristics. I like to think of the phenotype as the physical type. It's what we see. The genotype, an organism's genotype, is its genetic composition, and again, I think it's nice and easy to think of it as the genotype is the genetic type. So it's the actual letters, big A, little a, or big B, little b, big T, that it has. So an organism's heterozygous for a recessive allele, such as albinism, would exhibit the dominant trait. So if it's heterozygous, so if it is big A, little a, it's going to show the dominant trait. Yet it would possess the heterozygous genotype. So it would have a normal phenotype. Physically, we wouldn't be able to tell that it had a gene for albinism, but the genotype would be heterozygous for that. Okay, so what's the difference? Again, quick review between homozygous and heterozygous. So a homozygous trait is made up of two of the same alleles. So if we had an individual that was big R, the only gamete that it could produce would be a big R. But a heterozygous is one made up of two different alleles. So an individual that was big R, little r, could produce two different alleles. And if those two were to cross, so if you had a homozygous by heterozygous cross, then what we would end up with is we would end up with two individuals that receive the dominant big R gene from both parents and would remain homozygous dominant. But we'd have two individuals that would get the recessive allele from one parent and the dominant from the other. And we would have two individuals that would be heterozygous for that trait. So again, just keep those in mind as we go through. Homo, the prefix means the same. Hetero means mixed or different. Okay, so here's an example of the eye color. Okay, so blue eyes are actually the recessive allele little b. So the phenotype would be for blue eyes. That's what they physically look like, yet their genotype would be little b. Okay? Now brown eyes, there's your phenotype. Their genotype, because big B, the brown eye, is dominant, could be big B, little b, or big B, big B. So it could be either. So here, the same phenotype, might have different genotypes. So big B, little b look the same on the surface. So if you go up to someone and you see that they have brown eyes, you can't tell whether they are heterozygous or homozygous for that trait. You know that they have one dominant allele, but you can't tell which it is. But if you go up to someone who has blue eyes, you know right away the only way they can have blue eyes if there's homozygous recessive and they have two little b's. Okay, so here's an example with a color. So if we have two heterozygous individuals, so let's do a heterozygous, oops, I don't know what that was. Heterozygous, let's try this again. If we do a heterozygous mix, which means mixed, so if we do a heterozygous cross, someone with brown eyes, that's heterozygous. The gametes they could produce could be big B or little b. Same thing over here, the gametes of the second individual could be big B or little b. And if we go through and we do the cross, you'll see there should be one individual that is homozygous dominant, big B, big B. There should be two individuals that are going to be heterozygous, big B, little b. Now, all three of those individuals so far, or 75% of the total, are going to express the dominant trait. There's only one individual down here that gets the homozygous recessive, so there's 25% that would be recessive. And again, this goes back to the previous section where we talked about there being a 3-to-1 ratio of dominance to recessive when we do a completely heterozygous cross. And this is the reason why shown in something called a Punnett square. And if we go back to Mendel's work with his pea plants, here's the flowers. So the pea flowers either had purple or white. Do you remember which ones were dominant? The purple color was dominant. So the phenotype of 3 of those individuals is going to be purple, and only 1 is going to be white. So there is our 3-to-1 phenotype ratio. And over on the right it shows, well, what are the genotypes of those? Well, on average, 1 of them will be the homozygous dominant, 2 will be heterozygous, and 1 will be homozygous recessive. So in terms of the genotype, we get a 1-to-2-to-1 ratio. So the phenotype and genotype ratios are slightly different. We're going to kind of focus on the phenotype ratio. And remember, when we break up the phenotypes, the genotypes, we get the gametes. So these are specialized sex cells produced by each parent for sexual reproduction. So you get either the sperm or the egg. And you've got to remember that they are haploid. They contain only half the normal amount of chromosomes that are split in half. So they carry only 1 allele for each character. So if they're heterozygous for a trait, say big T, little t, they're going to produce either a big T or a little t. They can't pass both on to their offspring. And so just a quick review, you've seen this slide already, but Mendel's law of segregation says that the separation of alleles into separate gametes takes place. So if you've got individuals that are homozygous, either dominant or recessive, the only alleles they can produce are either big P or little p. But if you've got an individual that is heterozygous, it's a 50-50 chance that they will pass on either the dominant or the recessive. Okay, and here's just a schematic. If we were looking at an allele, call it A, the gametes produced are either going to be big A or they're going to be little a. They can't pass on both to one offspring. And here's another example of a Punnett square of that. So the gamete, remember, only has 1 copy of that. So the gamete alleles for a heterozygous are big B or little b, big B or little b. And again, we see that we have 3 of the individuals that will have the dominant trait, but only 1 of them has the recessive. So again, that gives us that 3-to-1 ratio that we've been talking about when we look at Punnett squares and we look at heterozygous crosses. And the last one, quick review. Again, you've seen this slide already, but the law of independent assortment says that there's independent segregation of pairs of alleles. So now, if we're looking at something called a dihybrid cross, so there are 2 traits. Those traits will assort independently of each other. So again, we can only have either a big T or a little t in a gamete. We can't have both. Same thing with the Q. So if we look at T and Q, the 4 possible outcomes of that would be big T, big Q, or big T, little q, and then little t, big Q, little t, little q. You can't have 2 Qs, you can't have 2 T's, you got to have 1 of each, but they're independent of each other. And so if we look at this example, so here's the maternal contribution, 1 of each, big A, big B. Here's the paternal contribution. So we get an individual that is completely heterozygous for those 2 traits. So they've got big A, big B, little b. So the gametes they can produce, there's 4 different ones. They could look like the mother, they could look like the father, in this case the grandmother and grandfather, or they could be recombinants. And here's where genetic diversity comes in that we talked about in the previous section. And the last one we'll look at, so here's Mendel's P's, so looking at 2 traits. So we've got yellow being dominant over green in terms of seed color, and then we've got the round shape being dominant over wrinkled shape in terms of the shape. So the alleles in the parental cell right here are big Y, big R, little r. So it's completely heterozygous. Now during gamete formation, those will independently assort, and the 4 combinations that we should get in equal ratios, a quarter each, are shown right here. So there's the 4 possible. Now if we go and we put those in a Punnett square, so if we take these 4 and we put them into one, the y-axis representing maybe mom and the x-axis representing dad, you can see when we cross, we get the combinations where we get our diploid individuals back. And just to kind of wrap this one up here, you can see that there will be 9 individuals that have both dominant traits. There will be 3 individuals that are round, which is dominant, but they're green, which is recessive. There will be 3 individuals that are yellow, which is dominant, but wrinkled, which is recessive. And there will only be 1, and it got cut off there, that would be green and wrinkled, the double recessive. And it's a little bit ahead of what we need to do right now, but that's where we're going to end up, going into what's called a dihybrid Punnett square. So again, thank you for your time.